Good afternoon, dear students. In the last lecture, we discussed about the Lac Operon, the structure of Lac Operon, the various genes that control the expression of Lac Operon, that is regulator gene, promoter gene, operator gene, and what are the three different structural genes. Okay, what we studied was there are three different structural genes, gene Z, Y, A. Together, all the three co-transcribe into a single mRNA, and then the proteins or the enzymes, that is beta-galactosidase, Lac permease and trans-acetylase, are synthesized respectively by Lac Z gene, Lac Y gene and Lac A gene. Okay, now there are promoter gene, which is the binding site of RNA polymerase enzyme. There is operator gene, which is the binding site of repressor protein. Okay, and this repressor protein is synthesized by the regulator gene. Okay, now today we are going to discuss about the regulation of gene expression, regulation of transcription of Lac Operon. Okay. There are different types of operon control mechanism or regulation of protein synthesis. Okay, the control of protein synthesis, the control of protein synthesis by regulator protein may be brought about by. Okay, what we are discussing today is how operon control is done, how regulation of protein synthesis takes place. Okay, so the protein synthesis, that is expression of Z, Y and A gene, okay, is controlled by regulator protein. How? By two mechanisms. Number one, induction or inducible system, and number two, repression or repressible system. There can also be two more types of mechanism, that is negative control of operon and positive control of operon. Okay, so Lac Operon, regulation by inducible system or induction, and repressible system or repression. We are going to study today the regulation of gene expression by induction and by repression. Okay, so let us first discuss what is induction. Okay, there are a category of enzymes which are called as inducible enzymes. Okay, these enzymes, that is inducible enzymes, are normally absent in the cell, or they may be present in very small quantities. They increase in number only in the presence of an inducer. Such a system is called as inducible system. Okay, once again, there are certain enzymes called as inducible enzymes which are normally absent in a cell, or they may be present in a very small quantity. But inducible enzymes are synthesized or their quantity increases only in the presence of inducer. Such a system is called inducible system. That means inducer is a molecule which increases the production of enzyme. Okay. For example, in bacterial culture of E. coli, growing in a medium containing glucose as a carbon source. Okay, so if E. coli is grown in presence of the sugar glucose, very small amount of or a very small quantity of beta-galactosidase enzyme is synthesized. Why? What is the role of beta-galactosidase enzyme? Beta-galactosidase enzyme breaks lactose into glucose and galactose. Okay, when organism is grown in presence of glucose, so there is no question of lactose, there is no question of breaking lactose. Okay, so enzyme is not required. That is why when E. coli is grown in presence of glucose, beta galactosidase enzyme is produced in very minute quantity because it is not required. Only one or two molecules of enzyme may be present. But when glucose is exhausted, okay, and when lactose is added to the medium, okay, when lactose is added to the medium, production of beta galactosidase starts, and within 2 to 3 minutes, about 3000 molecules of the enzymes are synthesized by Z gene. Okay, so as soon as lactose is added to the medium, suddenly, abruptly, there is a rise in the synthesis of beta galactosidase enzyme, and Z gene will produce almost 3000 molecules within 2 to 3 minutes. Why? Because now it will be required to break the lactose. Okay, beta galactosidase enzyme hydrolyzes lactose into glucose and galactose. Lactose is a disaccharide. It cannot directly enter into EMP pathway for energy generation, so it has to be broken down. So beta galactosidase enzyme breaks the sugar lactose into glucose and galactose. Some of the lactose molecules are converted into allolactose, and allolactose is an isomer of lactose actually. So Allolactose is the actual inducer for beta galactosidase synthesis. Okay, so lactose is an apparent inducer, but actual inducer is allolactose. Okay, in the lac system, allolactose is the internal or actual inducer, while lactose is the external or apparent inducer. Right? If you are studying this system in laboratory, then we can use a artificial inducer, okay, for our experimental work. And what is that artificial inducer? IPTG, isopropyl thiogalactoside. Okay, so it is a non-utilizable inducer. So if you want to study these type of mechanisms or regulation of gene expression in laboratory, then artificial inducer, that is IPTG, can be used. Right? So what happens in inducible system? First of all, inducer may be present or inducer may be absent. So let us discuss what happens when inducer is present. Say for example, the example I gave you, E. coli grown in presence of glucose, okay, will not produce beta galactosidase enzyme. That means, okay, and as soon as you add lactose, beta galactosidase will be synthesized. So in that case, lactose is actually the inducer. Okay, so the first case, inducer is absent. When inducer is absent, regulator gene produces active repressor protein. The active repressor protein binds to the operator gene and blocks the operator gene. When the operator gene is blocked, RNA polymerase that is bound to promoter gene cannot move forward. The RNA polymerase cannot reach up to structural genes, and hence no mRNA transcription and no enzyme synthesis. That means operon expression does not take place. Okay, when inducer is absent. We are talking about inducible system. Second case, inducer is present. Okay, again regulator gene will produce active repressor protein. Okay, but the active repressor protein combines with inducer. Rather, you can say that the inducer combines with the active repressor protein and converts it into inactive form. How? Inducer binds to the active repressor, brings about conformational change in the repressor and make it inactive. When the repressor protein becomes inactive, it cannot bind operator gene because it loses its structure, shape, configuration. Okay, so it becomes inactive. It cannot bind operator gene. That means operator gene is free. So RNA polymerase bound to promoter will move towards structural genes, and structural genes will be expressed. mRNA synthesis will take place, and enzyme and protein or protein synthesis will also take place. So according to inducible system, if inducer is absent, operon expression is off. Operon expression will not take place. If inducer is present, operon expression will take place, and all the three enzymes, beta galactosidase, permeases and transacetylase will be synthesized. Okay, so look at the diagram. The first gene, Lac I, which is written, is the regulator gene. It has produced an active repressor protein or regulator protein. Okay, look at this. Regulator gene produces active repressor protein, right? Now, the active regulator gene has produced mRNA, the active repressor protein. The active repressor protein, what it will do? Go and bind the operator gene. Why? Because it has affinity for operator gene. Operator gene is the binding site of repressor. So, repressor will go and bind the operator gene. Because the operator is blocked, the RNA polymerase enzyme, which is bound to the promoter, cannot move ahead. Okay. So, the RNA polymerase enzyme bound to the promoter cannot move ahead, and hence no protein synthesis will take place. Okay. So, no protein synthesis. Why no protein synthesis? Because regulator gene has produced active repressor, it has bound the operator, and RNA polymerase movement is blocked. So, no protein synthesis, right? So, this is what the case when inducer is absent. Okay. Now, look at this diagram where inducer is present. Again, RNA polymerase enzyme. Again, regulator gene has produced active repressor protein. Okay. And active repressor protein there is an inducer molecule that is allolactose. It joins the active repressor and brings about conformational change in the active repressor protein, and which cannot bind the operator gene. Is that clear? So active repressor is converted into inactive repressor by binding of inducer molecule. So this cannot bind to the operator gene. It cannot bind the operator gene. So RNA polymerase can move ahead, and Lac Z gene, Lac Y gene and Lac A gene, all of the three are expressed, right? So look at the diagram. Regulator gene has produced repressor protein. It is active, but inducer binds to it, converts it into inactive form. So operator gene is not blocked. RNA polymerase moves ahead, and all the three structural genes are co-transcribed into a single mRNA, and all the three proteins are synthesized when lactose is present. Repressor is inactivated, okay, and operon genes are expressed. That means the working of operon is on in presence of inducer. Now let us discuss about the second system, that is repressible system. Okay, I hope you understood about inducible system. What is the inducible system says? If inducer is absent, operon expression is shut down. No expression of structural genes. Okay, but when inducer is present, all the three structural genes are expressed, because inducer converts active repressor into inactive form by binding to the repressor and converting it into inactive form. Okay, so the next system is repressible system. Right. The synthesis of an enzyme may be inhibited by the product of metabolism, as we study in feedback mechanism. Okay, the synthesis of an enzyme may be inhibited by the product of metabolism. For example, in E. coli, the enzyme tryptophan synthetase, which brings about metabolism of tryptophan, is inhibited by tryptophan itself. Okay, that means tryptophan itself inhibits the tryptophan synthetase enzyme because it metabolizes tryptophan. Okay, the tryptophan synthetase enzyme is formed in the bacterium if the bacterium is grown in tryptophan free medium. Okay, because if tryptophan is present in the medium, it will inhibit the synthesis of tryptophan synthetase enzyme because it will metabolize tryptophan. Okay, the amino acid histidine also acts as a repressor. This process is called as enzyme repression. This is called as enzyme repression, where the product of the metabolic pathway itself inhibits the enzyme. So such a system is repressible system. What happens in a repressible system? The protein that is produced by the regulator gene is actually inactive. Okay, understand this. In case of repressible system, according to repressible system, the protein that is produced by regulator gene is an inactive repressor protein. It is called as aporepressor. The aporepressor needs the support of co-repressor molecule. The aporepressor combines with the co-repressor or effector molecule, and then it becomes activated. Okay, so when it becomes activated, it undergoes conformational change. It binds and blocks the operator gene. The protein synthesis will then stop. Okay, what happens in the repressible system? The protein that is produced by regulator gene is inactive. That is, it is called as aporepressor. The aporepressor combines with an effector molecule that is a co-repressor and undergoes conformational change, becomes active, then binds the operator and then blocks the operator, and operon expression does not take place. That means protein synthesis is stopped. Okay. So let us discuss about repressible system. What happens when a co-repressor is absent? Co-repressor is absent. Co-repressor is the effector molecule. So when co-repressor is absent, regulator gene produces inactive repressor protein, that is aporepressor. Because the repressor is inactive, it cannot bind operator gene. So it does not block the operator gene. If the operator gene is not blocked, RNA polymerase moves ahead. mRNA synthesis of the structural genes takes place, and hence enzyme synthesis also take place. That means here regulator gene produces inactive repressor. Inactive repressor cannot bind operator gene, and hence operator gene is free, and mRNA transcription and protein synthesis takes place. Operon is on. Okay, because co-repressor is absent. What happens when co-repressor is present? Okay, again regulator gene here produces inactive repressor protein, that is aporepressor. But because co-repressor is present, co-repressor will combine with the aporepressor, that is inactive repressor protein, and convert it into active repressor protein. Okay, so a co-repressor converts inactive repressor into active repressor. Okay, this active repressor can bind the operator gene. So it goes and binds the operator gene and blocks the operator gene. It blocks the operator gene means what? It blocks the movement of RNA polymerase. Okay, and hence no mRNA synthesis from structural genes and no protein synthesis take place. Okay, so according to repressible system, the effector molecule is the co-repressor. So if co-repressor is absent, no protein synthesis. If co-repressor is absent, operon expression will take place, enzyme synthesis will take place. But if co-repressor is present, enzyme synthesis will not take place. Okay, so let us understand with this diagram the upper one. Let us discuss about the first one. Okay, look at this diagram. So inactive repressor is synthesized by regulator gene. Okay, inactive repressor is synthesized by regulator gene. Because it is inactive, it cannot bind the operator gene. Okay, because it is inactive, it cannot bind the operator gene. So operator gene is free, and hence this RNA polymerase enzyme can move ahead. RNA polymerase enzyme moves ahead, reaches the structural gene Z, Y and A. Okay, and all the three structural genes are transcribed and protein synthesis takes place. Is that clear? So in the absence of co-repressor, when co-repressor is absent, all the three structural genes are expressed. Okay. Now the second case, when co-repressor is present, again the regulator gene produces inactive repressor protein, but co-repressor binds to the inactive protein and makes it active, and hence RNA polymerase cannot move ahead. And because RNA polymerase cannot move ahead, the expression of operon is blocked. Okay, that means structural genes do not produce RNA and do not produce any enzyme. So is that clear? So if co-repressor is absent, expression of genes will take place. If co-repressor is present, expression of genes will not take place. Okay, so that is about repressible system. So how operon is controlled, how regulation of gene expression or how regulation of operon takes place? By inducible system or by repressible system. Okay, so that's all about inducible and repressible system. Next time we will discuss about positive and negative operon control mechanism as well as role of cyclic AMP in operon.